In 2015, scientists described 1,500 fossils of a new human species with a brain 1/3 the size of yours, estimated at 2 to 3 million years old. They were wrong by an order of magnitude. 6 blind dating methods revealed that Homo Naledi lived just 236 to 335,000 years ago, alongside early Homo sapiens. The story begins inside a chamber no one was supposed to reach. In September 2013, Rick Hunter and Stephen Tucker navigated the Rising Star cave system in South Africa and located a fossil-rich chamber later named Dinaledi. The passage to this chamber included a vertical chute that narrowed to just 18 centimeters, and it required specialized caving skills. Lee Berger of Wits University assembled a team of 6 caving-trained scientists, known as the underground astronauts, to excavate the site. The chamber floor measured roughly 7 by 5 meters and contained a dense concentration of hominin bones within a silty clay matrix. Over several weeks, the team recovered 1,550 identifiable fossil specimens. Documentation included detailed 3-dimensional mapping of each bone's position, specimen labeling, and context sheets for every find. Taphonomic analysis established a minimum of 15 individuals based on repeated skeletal elements and size variation. The assemblage included skull fragments, jaws, teeth, vertebrae, ribs, limb bones, hands, and feet. Most fossils were concentrated in a central trough of the chamber's primary sedimentary unit, labeled SUA. Additional remains were embedded in thin silt layers and in microfractures of overlying flowstone plates. The entire collection was transported to the Evolutionary Studies Institute at Wits University and the University of Johannesburg for conservation and study. The rapid release of high-resolution chamber models and photographs in late 2013 and early 2014 drew international attention and set the foundation for subsequent anatomical and chronological analysis. The physical scale and logistical demands of the Rising Star excavation made it the largest and most systematically documented hominin recovery in African paleoanthropology to date. The first published analysis of the Rising Star fossils appeared in eLife in September 2015. Lee Berger's team, including John Hawkes and Paul Dirks, described a suite of anatomical features that immediately challenged established evolutionary timelines. The cranial capacity of Homo Naledi, measured from multiple skulls, ranged from 465 to 560 cubic centimeters. This value is closer to Australopithecus, or early Homo species, than to modern Homo sapiens, whose average brain volume is about 1,350 cubic centimeters. The skulls displayed a low, elongated vault, prominent brow ridges, and a pronounced occipital torus, traits found in hominins dating to 2 or even 3 million years ago. Yet the dentition showed a mosaic pattern. The anterior teeth were large with thick enamel, but the post-canine teeth were reduced and arranged in a parabolic arcade, a configuration found in later Homo. The mandible combined a thick corpus with a reduced canine, again mixing archaic and derived traits. Post-cranial bones deepened the puzzle. The hands included curved fingers, suited for climbing, but the thumbs and wrists were capable of the precision grip seen in modern humans. The lower limbs were gracile, with femur and tibia shapes supporting a fully upright bipedal gait. The pelvis blended an archaic iliac blade with a more modern sacrum. This anatomical mosaic did not fit any known hominin taxon. Lacking associated fauna, tools, or charcoal, the team relied on comparative anatomy. The small brain size and primitive cranial features led to an initial age estimate of 2 to 3 million years. This hypothesis placed Homo naledi among early Homo or even late Australopiths, intensifying the urgency to obtain direct chronological data. Standard dating methods proved unworkable in the Dinaledi chamber. No volcanic ash layers were present for argon-argon or fission track dating. The cave's dry environment preserved almost no organic material, no charcoal, and no collagen in bone, eliminating radiocarbon analysis. Fossil bones were partially encased in flowstone, but this carbonate crust posed a new problem. Flowstone can act as a chemical barrier, disrupting the uranium-thorium balance needed for accurate uranium-thorium dating. Groundwater movement in the karst system further complicated matters, as water could transport minerals and rework sediments, violating the closed system conditions required by most geochronological techniques. Stratigraphic correlation was unreliable, since repeated sediment disturbance and the absence of associated fauna or artifacts removed any external age anchors. These obstacles made it impossible to assign a credible age using conventional approaches. The only viable path forward was to design a multi-method campaign, applying several independent dating techniques to cross-check results and minimize the risk of error introduced by any single method or material. In early 2017, an international consortium led by Paul Dirks and including researchers from CENIEH, Wits University, James Cook University, and Griffith University initiated a blind dating campaign to resolve the age of Homo Naledi. The project divided samples from the Naledi chamber, including flowstone, tooth enamel, sediment, and charcoal, among 6 independent laboratories. Each laboratory received only anonymized sample codes and precise 3-dimensional provenance data, with no information about the fossil association or the results of the other teams. The methods applied were uranium-thorium dating of flowstone, electron spin resonance on tooth enamel, optically stimulated luminescence of quartz-rich sediments, paleomagnetic analysis of flowstone laminage, radiocarbon dating of charcoal, and a parallel uranium-thorium assay on tooth dentine. A central data manager at CENIEH maintained the chain of custody and integrated the age ranges after all laboratories submitted their results. The blind protocol ensured that no laboratory could tailor its analysis based on prior expectations or the findings of others. This multi-method, multi-lab approach was designed to eliminate confirmation bias and provide a robust, cross-validated chronology for the fossil assemblage. The final integration of all 6 dating methods was conducted using the Oxcal Bayesian modeling platform at CENIEH. Each laboratory's results, uranium-thorium, electron spin resonance, optically stimulated luminescence, paleomagnetism, radiocarbon, and a second uranium-thorium assay, were entered without prior knowledge of the others. The model incorporated stratigraphic constraints from the Naledi chamber, treating the flowstone as a cap above the fossil horizon and the sediment as the base. The resulting posterior probability distribution revealed a tightly bounded age interval. The 95% credible range falls between 236,000 and 335,000 years before present, with the highest probability density peaking near 285,000 years. This result, published in eLife in 2017 by Paul Dirks and an international consortium, overturned the earlier morphological estimate of 2 to 3 million years. The new chronology places Homo Naledi in the late Middle Pleistocene, overlapping with early Homo sapiens in southern Africa. The convergence of 6 independent methods, all cross-validated under a blind protocol, established this age as the most robust for any hominin assemblage in the region. Homo Naledi's confirmed overlap with early Homo sapiens in southern Africa reframes the evolutionary context of the Middle Pleistocene. Fossil evidence from Border Cave, Classis River Mouth, and Deep Kloof Rock Shelter places Homo sapiens in the region between 300,000 and 200,000 years ago, within the same interval established for Homo Naledi by the 2017 multi-method dating campaign led by Paul Dirks and colleagues. This contemporaneity raises fundamental questions about cognitive diversity among coexisting hominins. Homo Naledi's brain volume, measured between 465 and 560 cubic centimeters, stands in stark contrast to the average 1,350 cubic centimeters of Homo sapiens, yet both species display a mix of primitive and derived anatomical features. The persistence of a small-brained, anatomically mosaic hominin alongside modern humans challenges models that link symbolic behavior and cultural complexity strictly to brain size. The implications of this overlap set the stage for the scrutiny of behavioral evidence reported in the 2025 burial and engraving studies. In September 2025, a team led by Lee Berger, John Hawkes, and Patrick Randolph-Quinney reported a partial Homo Naledi skeleton in a shallow pit within the Naledi chamber. The articulated lower limb lay flexed and was partially covered by a distinct sand-rich sediment layer. Taphonomic analysis found minimal carnivore damage, limited disturbance, and sediment infill consistent with deliberate placement. The pit's dimensions, about 0.8 meters long, 0.5 meters wide, and 0.3 meters deep, matched intentional excavation rather than natural depressions. Reviewer commentary recognized the high-resolution sediment microscopy and contextual data, though some questioned whether sediment transport could mimic burial. In July 2025, a separate group described geometric engravings on a quartzite slab near the fossil horizon. The grooves averaged 0.9 millimeters wide and 4.2 millimeters apart, forming a rectilinear grid. 3-dimensional laser scanning and experimental replication with stone tools supported intentional marking, but critics argued for possible natural abrasion. Both findings remain under active debate, with authors cautioning that evidence for symbolic behavior is not yet conclusive. 2 competing phylogenetic models continue to divide researchers. One positions Homo Naledi as a late surviving branch of early Homo, while another aligns it closer to Australopithecus, based on distinct cranial and postcranial character matrices. The Naledi chamber, discovered in 2015, holds over 150 additional Homo Naledi specimens, including a nearly complete cranium, but remains undated due to the absence of suitable flowstone or charcoal. Direct age estimates for Naledi are pending, with future work likely to rely on non-destructive techniques or paleoproteomic analysis. Advances in protein sequencing may soon clarify both the evolutionary placement and the temporal range of the species. The search for associated tools, fire residues, or further symbolic artifacts remains ongoing. Each new discovery will test the limits of current models and drive the next phase of research. Since the 2013 Rising Star discovery by Rick Hunter and Stephen Tucker, and through Lee Berger's 2015 species description, the 2017 blind multi-laboratory dating led by Paul Dirks, and the 2025 burial evidence, one fact remains: Homo naledi's behavioral repertoire, including tool use, fire, and symbolism, remains unresolved. The search for answers now defines paleoanthropology.